Hi, welcome to its Biochemistry, and in this lecture we'll be discussing the ionization of amino acids, and we'll go on to calculate the pi value of some of the amino acids. Now, what is an amino acid? It's a micromolecule having an amino group and a carboxylic acid group attached to the central alpha carbon. It'll be having a hydrogen atom and a side chain, an R group, and the characteristics of the amino acid will depend on the R group. Now, this amino acid can be ionized, which means both the amino group and carboxylic acid, both of them can be ionized, and both of them are ionized in the ionized form. It is called a Switter ion, or it is in the dipolar form. Let us see how amino acids are ionized and what are the characteristics of ionization. Now, Zwitter is a German word for hybrid, and obviously this Switter ion is having both positive and negative charges on it, so that's why hybrid. And Zwitter ion is considered as electrically charged. At the same time, it is electrically neutral also, since it is having both negative and positive charges. And so the net charge on the Zwitter ion is 0. And amino acid in Zwitter ion form reacts both with the acids and bases, and it shows what is called amphoteric behavior. In chemistry, amphoteric compound is a molecule or ion that can react both as an acid and as a base. So that is why amphoteric nature. And amino acids are also called ampholytes. Now, ampholytes, which are compounds that when dissolved in water, so that is dissolved in water, they can act either as acid or as a base. The point at which the Zwitter ion has no net charge is called isoelectric point. And the pH at which this happens is the isoelectric pH. And at this point, the amino acid is considered as electrically neutral, as I said, and the solubility will be minimum. And hence, there is no mobility in electric field, which means that it won't move during electrophoresis. And at physiological pH of 7.4, most amino acids exist as Zwitter ions. Now, let us consider a condition when we add an acid, for example, HCl, to this Zwitter ion solution of amino acid. Now, practically what we do is that the pH is taken to a very low level, and this ionized carboxylic acid group gets protonated at this condition because H plus is available in plenty, and the whole molecule becomes a cation. And on the other hand, if we add a base, for example, NaOH, to this Zwitter ion solution, the carboxylic acid group remains the same, remains ionized, whereas the amino group gets deprotonated. The H plus is lost to the solution, and the whole molecule becomes an anion. And here there are two values, pK1 and pK2. Let us discuss about this pK1 and pK2, taking ionization of glycine as an example. So this is a case when the solution is having a very low, kept at very low pH. The pH is 1.0, and the glycine, the cationic form of glycine here, glycine exists almost entirely as cation form at this pH of 1.0, with a net positive charge of positive 1.0. And we add to the solution, there are plenty of H plus ions, obviously. We add NaOH, the base, slowly, and slowly. Now, the carboxylic acid group of this cation starts to get deprotonated. H plus is progressively lost from this cation. And at a pH of 2.34, there will be an equal mixture of glycine cation and the Zwitter ion of glycine, with as you can see, with the deprotonation of this group, carboxylic acid group. And there is an equal mixture of cation and Zwitter ion. That means the average or net positive charge is plus 0.5 now, which also means that 50% of the glycine is in cation form and the rest 50% is in Zwitter ion form in the solution now. And as we continue to add NaOH, the base in the solution, what happens is that this group, amino group, also gets deprotonated. deprotonated. And at a pH of 9.6, there is an equal mixture of Zwitter ion and this glycine anion with the loss of H plus from this amino group. And the average net positive charge now is plus 0.5, which means here at this point, there will be 50% of Zwitter ion as well as 50% of anion. And beyond this pH value of 9.6, when concentration of anion overtakes the Zwitter ion concentration, the charge tends to become negative, more negative, and it reaches -1 when glycine almost entirely exists as anion here, -1. Now, these two values on either side of Zwitter ion, 2.34 is taken as pK1 value, and this value 9.6 is taken as pK2 value. This is actually pK1 value with respect to carboxylic acid group, and 9.6 is pK2 value with respect to amino group. Now, the sign and the magnitude of the net charge of any amino acid at any pH can be predicted in the same way, just as we discussed or analyzed the ionization of glycine. Let us calculate the Pi value of glycine from the data that we have. The Pi of glycine is equal to pK1 plus pK2 divided by 2, which means 2.34 plus 9.6 divided by 2 is 5.97. And it has been found that the best buffering action of amino acids occurs at pK1 and pK2, and the least buffering action at Pi value. Now, let us examine the pK1, pK2 and the Pi values of the other amino acids, the other proteogenic amino acids. All the 20 amino acids are shown here. You can see here. Now, here there is a new value, that is pK3 value, which can be seen in the case of acidic amino acids and basic amino acids. Here aspartic acid, glutamic acid, and these are the basic amino acids. And cysteine, some more amino acids like cysteine also shows a pK3 value that can be discussed in another lecture. Let us take another example of histidine, how histidine ionization is happening. Now, here, as in the case of previous example of glycine, here also at the start of the solution, the entire solution is at very low pH, and the entire histidine is in cationic form, with the ionized primary amino group, ionized this R group, and a protonated carboxylic acid group. And as in the previous case, we add slowly NaOH progressively at the start of the experiment. The solution is having high concentration of H plus ions, and NaOH is added slowly. So, what happens is that like this. At first, this COOH group gets deprotonated. At this particular pH, it will be 50-50, and at pH of 6, what happens is that the R group gets deprotonated, and further addition of NaOH causes, so this is obviously the Zwitter ion. You can see that the amino group, the primary amino group, and the primary carboxylic acid group, both of them are ionized, and this is the Zwitter ion form, and further addition of NaOH gives deprotonation of this primary amino group also. So this again at this point will be 50% Zwitter ion and 50% of entire anion of histidine. Now here the pK1 value is 1.8 with respect to carboxylic acid group, and pK2 value is 9.17 with respect to the amino group, and the new pK3 value, so this is actually the pK3 value, it is 6.0 with respect to the R group, the side chain amino group on the R group. And the values on the either side of the Zwitter ion, are taken for the calculation of Pi value, just as in the case of glycine. In the case of glycine also, we had taken the values on either side of the Zwitter ion. So what is the Pi value of histidine? That is pK2 plus pK3 divided by 2. Now It is 9.17 plus 6.0 divided by 2, it gives 7.59. You can see here is the value, and obviously in comparison to other amino acids, this 7.59 value, the pi value of 7.59 has got a significance. With the pi value of 7.59 and pK3 value of 6.0, histidine forms the best amino acid or the only amino acid with the pH 7.4, the physiological pH of 7.4 within its buffering range. So that histidine forms the best amino acid buffer under physiological conditions also, and that's why histidine contributes to the buffering capacity of plasma protein and hemoglobin under physiological conditions in our blood buffers. With the pi value of 7.59 and pK3 value of 6.0, histidine forms the best amino acid or the only amino acid with the pH 7.4, the physiological pH of 7.4 within its buffering range. So that histidine forms the best amino acid buffer under physiological conditions also, and that's why histidine contributes to the buffering capacity of plasma protein and hemoglobin under physiological conditions in our blood buffers.